Hi, it's Elodie from Biology 1951, Section 3. This is my role model project on Siddhartha Mukherjee. So he was born and raised in New Delhi. He first fell in love with life sciences from having an amazing biology teacher who taught him a lot about cells, and he realized he wanted to do that more, deciding to pursue it for his undergraduate studies at Stanford. He read a lot of books during his time there, including some by other scientists, which led him to take an interest in medicine after he realized that you don't just have to work with patients as a doctor, you can also make discoveries. He then went on to Oxford University to study immunology for his doctorate, then Harvard Medical School for an MD in oncology. He then began work finding it both exhilarating and taxing, because of course he's having to deal with cancer patients who are suffering, and then also many of whom die. As he continued to ask questions about cancer and ethics, he decided to take up writing, documenting his thoughts, and then writing many books. Some of his hobbies include singing, including when he was young, he sang ragas, and then he began singing again in college at Stanford, then gave it up and took it up again a few years ago, saying it helped him find his voice as a doctor. He also likes gardening with his daughters and cooking with his wife. What I admire about him, we both love seeing things from the perspective of cells. I read his book, Song of the Cell, really loved it. That's actually what got me into biochemistry, just because the way he explained all these different kinds of cells, and like his own work made me think I want to do that too. And so now here we are. Cancer, we're both very interested in how cancer cells work, particularly how they differ from regular cells. Ethnic, we were both Indian, and we both love to write. His relation to biology, so like his relation to our class, he is a doctor, so he's dealing with biology every day. He specializes in the study of cancer as an oncologist. Specifically, he does a lot of work with blood cancers, so leukemias. So the research article I chose was on cancer-associated fibroblasts. The question addressed was, what are the origins and contributions of colorectal cancer-associated fibroblasts associated with poor prognosis? So, like, you don't have much longer left to live. That's what prognosis is, or poor prognosis. CRC, so colorectal cancer organoids, were injected into the colons of knockout mice so they could observe how the CAFs contribute to the start of cancer or tumorigenesis. How experiments were conducted in knockout mice, so mice where some genes were shut off. Control used, they injected a different chemical. So MCAM, hemagglutinin. MCAM is a melanoma cell adhesion molecule that binds to hemagglutinin. And so the control they used was having another hemagglutinin with a different thing binded to it to see if macrophages respond the same way to all hemagglutinin or just the ones that MCAM bind to. So here's the data figure. This part is the most important part, so I'm going to be talking about this. These are the fibroblasts. This is what it looks like inside the cells. The IL-1 beta ligand connects to the receptor, signaling for phosphorus to connect to P65, which then binds to DNA as an activator to signal to produce RNA for these two proteins, which are then released outside the cell and signal for macrophages to come in. Not the ones that like destroy the pathogens, but the ones that help the injured cell at like a cut site or something. Helps the injured cells by like giving them resources and like dampening the inflammation. So they come to the cancer cells and basically are just like dampening down inflammation, saying this is not a problem, this tumor is fine, while also giving it resources and feeding it. So conclusions, in CRC, CRFs are produced by intestinal paracryptoleptin receptor cells, which then produce MCAMs, a CRC stroma-specific, so stroma-specific basically meaning fibroblasts, marker. High MCAM expression lowers patient survival for humans, as more macrophages, dampening immune response and promoting tumor growth, as there are more. So basically just more CAFs, more MCAMs, more macrophages, meaning more fuel for cancer. So discoveries made by Mukherjee. He did some research on cancer cell evasion, so how cancer cells hide. They evade the immune response by hiding behind healthy cells. He's done work in immunotherapy, such as the work with Emily Whitehead, as we explored in that one article. He also has done a lot of, he did a tumor study. So he researched how tumor-associated neutrophil precursors how they limit DNA repair, which is originally the article I was going to do my whole explanation on, but it was way too complicated, so I ditched that idea. And then also osteoarthritis, so how the loss of certain cartilage cells causes osteoarthritis. So next up, he's currently working on studying how eating habits may influence our risk of cancer and AML, acute myeloid lymphoma. So here are my citations. These three little symbols mean that not all authors are listed, because the thing I was using for my citations wouldn't write down all these authors' names, and there's like 50 of them, so I wasn't going to do that by hand. Primary sources, so these are the two ones I got my main study from. Here are the two ones I tried to do and then didn't work out, but I still used here. And then popular sources here and here.